So welcome everyone. It looks like our numbers have stabilized a little bit here. I'm really pleased that you could join us today on this beautiful Saturday afternoon. I'm Autumn Sabo. I'm one of the 11 PA Botany committee members who have worked hard to deliver you today's symposium. So thank you for being here. As we get started, I just wanted to tell you a little bit about what PA Botany is, in case this is your first time with us. So PA Botany is an all-volunteer outreach and networking effort. It started in 2010 when there were a group of dedicated and passionate botanists who came together because of a concern over a decline in resources and college programs that were focusing on botany. So our hope with PA Botany is that it will inspire other people to appreciate our native flora, and that together we can all sort of bond over our love of plants and help to know more and more about PA's botany. This is the 5th symposium bringing us all together. This is our planning committee. As I mentioned, we have 11 members from across the state. We also have been working in close partnership with the Natural Areas Association to make this symposium possible. If you have any questions about what PA Botany does, or if you are interested in joining us in a greater capacity, please put your name in the chat and one of us will reach out to you. In addition to our committee, we have gotten a lot of support through the years from Western Pennsylvania Conservancy. They have been our fiscal sponsor throughout the years. Sheets Chewy Foundation has helped to underwrite both our webinars this past year as well as our symposium. So this has allowed us to have optional registration fees this year. We are thrilled to have all of you here. I really appreciate your involvement. And thank you for folks who have decided to donate through those optional registration fees. I mentioned that we are in partnership with the Natural Areas Association. They have been providing crucial programming and administrative services to the PA Botany Committee. We would not have been able to pull off this symposium without their help. They have also been key in helping us do PA Botany webinars throughout the year, which I'll mention a little bit more about soon. Natural Areas Association is a partner organization that advocates for botanical sciences, and that's because the botanical sciences are such an important part of natural areas management. Natural Areas Association has a webinar coming up soon that might be of interest to you if you want to learn more about how your plantings can help wildlife. You might also be interested in NAA's 49th conference coming up next year. Robin Wall-Kimmerer is the keynote speaker. She is the author of Braiding Sweetgrass and an expert in traditional ecological knowledge. If you need more reason to attend, the conference is going to be held in Duluth, Minnesota. This is a really lovely city, and the rose garden that overlooks the city and Lake Superior should still be blooming at that time. I mentioned that we've been having some webinars going on this past year. So they are on demand. You can still view them even if you weren't able to get them when they were going live. So please take a look at those if they might be of interest. These are kind of our substitute for some of our hands-on workshops that we normally have done. We do plan to have our PA Botany Symposium back on that regular 2-year schedule in person next November. So hope to see you all there at the Penn Stater for hands-on workshops and a lot more keynotes and student involvement. Okay, so now getting to what we all are really waiting for, right? Today's agenda. So we have a keynote by Dr. Peter Zale. We'll take a little bit of a break to get folks, moving on to the poster session. The student poster session will be in a different platform, so a different link to get you there. After you have an hour to view those posters and discuss the posters back and forth through comments on that page, then we will come back to this Zoom room for cool finds and the awards. So just a nice short day, 1 to 4. And even though we are virtual, we still want to hear from you. Please participate. We have the chat open. That's a place for you to post questions and comments for our speakers and other attendees. We will have the poster session, and you can comment and talk about that with students and other attendees in their comments. And then we will have a little bit of trivia every once in a while, just some little Zoom polls. If you need help at any time, we do have folks from Natural Areas Association waiting to help you. So you can just send a quick email to pabotany@naturalareas.org, or you can type in the chat if you need help sort of moving between all of these different facets. It is my pleasure to introduce Dr. Peter Zale. This is my first time meeting Dr. Peter Zale, but I've heard a lot about him. So I'm really excited for his talk today. He has studied plant breeding and genetics at the Ohio State University. And now he's been working at Longwood Gardens for a while. Hopefully we've all visited there, such a lovely place. His work there focuses on temperate terrestrial orchids from all over the world. He has also been working on building his own private botanical garden with great collections of native plants and hardy orchids, because he believes that we can use our gardens as a tool for plant conservation. So again, if you have any questions for Dr. Peter Zale, you can put them in the chat. We'll sort of save all those questions up, and Peter will answer those at the end of his presentation. So just one little poll to get us warmed up thinking about orchids. You're all so much better at participating than my students. I'm so proud of you. We have 85% participation already. So I'm gonna go ahead, 88, well done. So most of you have participated, awesome. I'm gonna go ahead and end this poll now. Up to 91%, super exciting. So you can see that most folks guessed that we had between 11 and 30 orchids. We actually have approximately 58 species of orchids. So that's according to Alan Weekly's Flora. I'm sure, given differences in taxonomy, we could count that differently depending on, which experts we use, but approximately 58 orchid species. I think that's just incredible. Okay, and with that, now I'm going to hand it over to talk to Dr. Peter Zale.
All right, thank you, Autumn, and thank you to the committee for having me speak. I'm just going to share my screen. Okay, everybody can see me and hear me and see the presentation okay? Great. All right, thank you everybody for having me. I'm really excited to be here and to talk about what we've been doing at Longwood. And so I'm just going to dive right into things here. I think most people, when they think of Longwood Gardens, probably think of something like this, like our 1,000-bloom chrysanthemum, which is typically on display at this time of the year, or maybe an orchid show that you might see in the winter time, or if you're here in the summertime, the Victoria pools. And then coming up is Christmas, which is really the busiest time of year at Longwood. So I think people tend to associate Longwood with, being a display garden and having these seasonal programming events and things like that. And that is really a big part of what Longwood is. But I'd like to take a step back and talk about the origins of Longwood and sort of set the stage for what I'm going to talk about today. And so long before Pierre DuPont, who is Longwood's founder, founded Longwood Gardens, the original farmers that owned this property was the Pierce family, a Quaker family that bought the property from the William Penn Commissioners. And George Pierce, the original owner, his twin great-grandsons really developed a love for trees. And over a several decades period, they planted all of these trees in what became known as Pierce's Park. And this really, became the foundation of Longwood. And so over the course of a couple hundred years, the trees were planted and curated, and then Pierre DuPont bought the property to save the trees. And really, I think that the story of Longwood begins as one of conservation, because had he not come in and bought these trees, they would have been cut down. There were already sawmills cut up and or already sawmills set up to cut down the trees. And so that's when Longwood Gardens started. And then many people are familiar with the story of Longwood, that over the years, the DuPonts built these great conservatories to house orchid collections like you see here, and built these wonderful display gardens and things like that. But if you step back in time a little bit and look at the plants that the DuPonts were really interested in, it's really quite fascinating. And so we know that they had this interest in tropical orchids and, orchid hybrids and things like that. And that's in large part why they built the conservatories that we have here at Longwood. But they were also founding members of the American Orchid Society. So they had this very broad interest in orchids. And we found through a project we did here called the Hagley Project, where we basically compiled all of the invoices and letters and correspondence that Pierre and Alice DuPont made with the people they were acquiring plants from. We found that out of 38,000 items, let's say, that they brought to Longwood Gardens, one of the first orchids that they bought was Galliera spectabilis, so which is really still a relatively locally common species here in southeastern PA. And so about 5 years ago, we were, well, 6-7 years ago now, we were interested in developing a plant conservation program, and this knowledge sort of came to light. And we started thinking about different ways that we could think about our orchid collection here, and to think about how Longwood could play a role in plant conservation. And this is something that really a lot of public gardens are doing now. A lot of public gardens are very conservation oriented, whether it's working with local species or species from around the world. And we wanted to enter that arena as well. And so we set up a conservation program really focused on native orchids. And you can see all the things that we're trying to achieve here. Number one is to help prevent the loss of local orchid diversity. Here in southeastern PA, the habitat is really fragmented and developed. And if you look at the herbarium record, this was historically a really rich area for orchids. Most of that appears to have been destroyed now. And so we're interested in sort of using horticulture to maybe bring some of those species back here, and really looking to see what the role of horticulture is in plant conservation. And if you move across look at different botanical gardens across the country, you see things happening, but we wanted to make a contribution to the public garden community and the scientific community through developing conservation horticulture. And out of this, we can build orchid collections here at Longwood that are not only beautiful, but have conservation value. And this allows us to dispel, I think, a lot of horticultural myths, like native orchids are impossible to grow, or you can't grow them without certain fungi and things like that. And so out of that, we've made a program that's really focused on learning how to propagate native orchids. And so we've worked very broadly with many species, looking at seed propagation, and then not only that, but how to grow them from that point. How do we grow them in our lab? How do we take them out of these very controlled conditions in the lab and then use them for displays and for restoration plantings here on the property and beyond, and then also for sending to other public gardens. Native orchids are not frequently cultivated at public gardens, and so it's really important for us to be able to share some of the plants that we're working with to help build other collections, and also as an ex situ conservation activity. So we do this, we've got a lot of great long-term partnerships for some of these activities, and I'll talk a little bit more about this. And most of our projects tend to be taxon based. And this project, in addition to sort of the legacy of orchids here at Longwood, is also an outgrowth of our plant exploration program. And so over the last 60 years or so, you can see all those dots represent places where Longwood staff has gone and collected non-native plants and brought them back to the gardens and used them for various things. And when I started here, those green dots on North America were non-existent. And so there's, beyond the world of orchids, a big push to really focus more on utilizing the native flora here at the gardens for display and for restoration purposes. And so this program is really a corollary to all the work that's happening there. And so if you look at the orchids of Pennsylvania, I know Autumn mentioned that there were 59 taxa in Pennsylvania, and we can always debate this based on what experts you decide to follow. I counted 61 based on the description of some new spiranthes here recently, but all of the orchids here are terrestrial, right? We don't have any epiphytic orchids like you find in the tropics. And I don't know if this is particularly accurate, but I would say that at least 23, probably more, are listed or threatened in some way. So, you know, I always say roughly half of them are rare in the wild. And if you look at their presence in public gardens, and that's partially what this table is showing, you don't really find native orchids growing in places like Longwood Gardens or, you know, other public gardens that you might visit here in the greater Philadelphia area. So we're really looking at, you know, how can we play a role in native orchid conservation? And one way we can do that is through some of the things I mentioned and through collections development, and really teaching people about the diversity of native orchids that we have here in the mid-Atlantic and really throughout the United States. So I'm going to talk about a couple of case studies. We're doing a lot of work here, and it's really impossible to present about all of it, but I wanted to use a couple of familiar things and really talk about where the project started, and that was with the genus Cypripedium, which, you know, if you're a native plant lover, is probably a genus well familiar to you. And from my perspective, it really is a flagship genus of, you know, with, you know, flagship species for conservation. You know, something like the yellow lady slipper is recognizable, it's charismatic, it's beautiful, and it's, again, it's a plant that, although not that rare, is not as common as it used to be. So we started with a population. At the time when we started this, we knew about one population of the large yellow lady slipper in Chester County, here in Chester County, Pennsylvania. Now we know about 3. And we wanted to work with the Brandywine Conservancy to learn how to propagate that. It's a very small population, only about a dozen plants. And so we started to learn about how we could do this. And we found some great information that came out of what's known as the Kew Gardens Sainsbury Orchid Project, where basically they had one lady slipper plant of their native lady slipper, Cypripedium calceolus, left in the wild, and they wanted to learn how to propagate it. And what they figured out was that embryo culture worked really well. And we know that this works for our native yellow lady slipper. But what's interesting is that if you look at sort of lady slipper orchids, large yellow lady slipper orchids in cultivation in the United States and at public gardens, most of them originated under that red dot right there, which is roughly on top of Itasca County, Minnesota, which is the headwaters of the Mississippi River and orchid paradise, and also the home of a gentleman named Bill Steele, who has basically dedicated his life to propagating Cypripediums and other native orchids and sending them out to people who are interested in growing ethically propagated plants. And so Bill has sent yellow lady slippers all over the country. And I've been up there to visit Bill, and he's doing phenomenal work and has been for a long time. But he's sending out mainly yellow lady slippers that are of northern Minnesota provenance. And if you look at the yellow blob on the map, that is basically the range of the species. And so you have this very widely distributed species with a lot of different taxonomic entities that are debated. And so we really wanted to focus on growing yellow lady slippers that came from our backyard, essentially. And so this slide is a bit dense, but what we basically do is to propagate these things is to hand pollinate them. We find that many native lady slipper orchids and other orchids seem to be pollinator limited, and so don't often set seeds of left on their own. So we hand pollinate them. We harvest the seeds about 50 days after pollination. And then we take those developing seeds and we put them onto tissue culture medium in our lab. So a sterile medium that was developed especially for lady slipper orchids with the key ingredient of russet potato. And so orchids have different requirements depending on what genus or what species they are. And so about 100 years ago, these researchers figured out that lady slippers like potato, other orchids like coconut water or pineapple. And so it's this really curious sort of black box to us, but we know that it works. So russet potato is added to the medium, and then we put them in our cabinets and incubate them in the dark. And they germinate within about 1 month, and roughly about 15 to 18 months after that, they're ready to go. And so this idea of embryo culture is basically harvesting immature seeds at a point when the embryo has developed, but the factors that contribute to the dormancy of that seed that make them difficult to germinate have not developed yet. And so the timing of doing this is really critical. But we have found for Cypripedium that 45 to 55 days after pollination works really well. And we've now worked across all native North American species of Cypripedium and some exotic species as well. So to give you an idea of how this works, chronologically speaking, so we would hand pollinate the flowers in early May. Towards the end of June, we'd harvest that partially developed capsule, and you can see in the image the spent flower still hanging on there. And then basically we surface sterilize that green capsule, put it into our sterile working space, scrape those seeds out and put them into jars or petri dishes. And within about a month, they start to germinate. And then within 4 or 5 months, they start to grow into little plantlets. They produce little shoots, they produce roots. And it's at that point that we move them into test tubes, such as you see here, and we grow them for about another month. And so at this point, they've grown about as much as they can. And what they need is cold. So we take them out of these test tubes. We take them out of, away from their nice medium that they've, been growing so well on, and we put them in the fridge and we keep them there for 4 months, because basically unless they get that cold period, they cannot grow anymore. After that time period, we plant them into pots, and as you can see here, and they produce their first shoot, and then they're ready for life outside of the test tube. And this is really, if you're growing orchids, the most, in our experience, and I think many others as well, the most difficult part of growing these plants is to get them from these nice sterile conditions in the lab to growing out in the nursery, in the gardens, etc. And if you grow them right, Cypripediums are really incredible plants. And large yellow lady slipper, if you're a gardener, there's several places online where you can purchase seed grown plants of these. In the right conditions, it's essentially, it's a very, very long lived plant, very beautiful, blooming around Mother's Day. And what's really interesting is that we find that the yellow lady slipper, which is one of the more common species in Pennsylvania, here you see a scene from Berks County with some really large clumps of yellow lady slippers. We find that it's one of the most difficult to transition from the lab to soil. We lose a lot of them, but the ones that survive do extremely well. And they tend to, just grow and make big clumps. And recently I was reviewing some literature that said that the European yellow lady slipper, the estimated generation times for that species are roughly 110 to 380 years. And so, even though there's this big sort of bottleneck to get the plants out of the test tubes and growing, we feel like the ones that do grow are going to be here for a long time. So, thinking about that, lady slippers actually make really great plants for restoration projects, because if you get the conditions right, they have the potential to live for a very, very long time. And so, taking the knowledge that we learned with the yellow lady slipper, we started another project with the pink lady slipper, which really is the most common species in Pennsylvania, but it's become locally rare. And so a few years back, there was a Grid magazine article that talked about the importance of digitizing herbarium specimens for accessibility. And in the article, the specimen you see there of the pink lady slipper was used basically to say that, the reason this is important is because we know that the pink lady slipper used to grow in the Wissahickon Valley, but it's not there anymore. And right after that was published, people started to come forward and say, oh, well, we know where the plants are. And this really snowballed into a project where we're working with the Wissahickon Garden Club, the Morris Arboretum, and Springside Chestnut Hill Academy to propagate these plants and enter them back into some restoration study sites. And so that's exactly what we did. The population in the Wissahickon is very small, just a few plants. We hand pollinated them in April. We went through our typical procedure of pollinating them, and they grew really well. And then what we found was when we transitioned them to medium, we got a lot of seedlings, as you can see in the center here. I don't know if you can't see my cursor, but if you look in the middle of the image there, there's seedlings popping up with white leaves. And so we think that might be partially due to inbreeding, due to the very small population size, but the plants seem to be growing well. And just recently, we worked with senior high school students at Springside Chestnut Hill Academy, who owns a little bit of property bordering the Wissahickon, and we found an area where we think that the pink lady slipper might grow. And so with students from one of the biology classes, we planted plants onto the property there, along with some native companion plants such as mountain laurel and vaccinium species. And so we are hoping to use this site as sort of a long-term study to see if what we're doing is really feasible for restoring populations of orchids. And so we work with all native lady slipper species. And the one I'm really most excited about is the Kentucky lady slipper, which is not native to Pennsylvania, but it's the rarest lady slipper in the United States. And we're using this species as a model to learn how to develop ex situ collections, genetically representative ex situ collections, so that, if native populations are extirpated, we have a sufficiently large and genetically diverse collection in cultivation that serves as a representation of what you would find in the wild. And so we're excited about this because this is a rare species, a globally rare species, vulnerable species. It is rare in every state of occurrence. And again, it's considered to be very difficult to grow. And so we've taken what we've learned. We've been able to apply that. We now have populations from Georgia and Texas and a really odd population in Virginia. And what we're scheduled to do is, like the pink lady slipper, is send a bunch of Cypripedium kentuckiense that we propagated from Texas back to the Forest Service there. And us and them, in conjunction with Stephen F. Austin University, want to plant some of them back into the wild to again see if this is a suitable restoration practice that really helps promote the long-term genetic health of these orchid populations. And so the first population we worked with was in Virginia, and this population is not on the radar of most people, and it's a really, really cool area. Just to, because I know a lot of people are out and about looking at plants, but you can see the lady slipper in the middle of the image there, and it's growing in a really rare habitat, this coastal plain basic seepage area. And so just wanted to give you an idea of where it grows. And Cypripedium kentuckiense, despite its really rare status in the wild, for us has been the easiest of all of the native lady slippers to grow. And so we've now been able to propagate these plants to the point where they're not only flowering, but we have them in the gardens. And so a couple hundred plants have been planted into Longwood, and you can see these on display now in the Pierce's Woodlands area and in the Forest Walk. And so that's something we're hoping to do more and more of, not only with Kentucky lady slipper, but yellow and pink and others native to Pennsylvania and as well. And so we have a saying at Longwood that it's not done until it's overdone. We, like big displays and, things that are really attractive to people. And so sort of as a model for all of this lady slipper work has been a company in China, Hengduan Biotechnology Limited, and they recognized that poaching of Cypripediums in China was a big problem. And so they started propagating them, and you can see in the images here that Cypripedium tibeticum, but we're trying to mimic what they're doing and propagate these plants in really large numbers so that we can use them for all different kinds of things. And so switching gears a little bit, so that gives you a bit of an overview of what we're doing with lady slippers, and something I'd like to talk about that sort of came out of all that work with lady slippers is work we've been doing with Platanthera. And we're really excited about Platantheras. Obviously, some of them are very beautiful, some of them are very rare, some of them are extirpated from Pennsylvania, and it's a very large genus worldwide with a good representation here in the US. And if you look at eastern species of the genus Platanthera, basically all of them are of concern in at least one place where they occur. And it does seem like there's been no red listing efforts, and I'm not, perhaps others can say otherwise, but it seems like a genus that seems to be particularly adversely threatened by invasive species and deer browse. And so populations seem to fluctuate widely, but also generally seem to be declining for many species throughout their range. And so this is a group we wanted to work with for various reasons. Conservation value, many of them are also very beautiful, and you don't find them in the nursery trade, or at least not plants that have been ethically propagated. But one thing that we know is that these are very, very difficult to propagate from seeds. And so we started doing a bunch of field work, just to show, here's iNaturalist, shows a little bit of the places that we've been focusing on looking for Platanthera. But if you look at seed germination literature for the genus Platanthera, really not much is known, and many species haven't been worked with, and those that have been worked with, there haven't been a lot of really positive outcomes. And so we tried to sow a lot of seeds of things like Platanthera paramina in the image there, and other like Platanthera grandiflora and related species with not much luck at all. Some of the other species are a bit easier, but these paramina and its allies are really quite difficult. And so we decided to focus on Platanthera paramina in conjunction with our friends from Pennsylvania DCNR and the Game Lands Commission, and work with a population near Harrisburg. And so Platanthera paramina, I'm sure is familiar to many. It's a PA threatened species. It seems to be, if you look at the BONAP maps and look at its various listings around the country, it seems to be rare in most of the places where it grows, and it's closely allied to very rare species like the federally endangered Platanthera leucophila and praeclera, and the debated Platanthera shriveri. But it's possible that if we are able to use a species like Platanthera paramina as a model and figure out how to successfully propagate it, then this information could be applied to these rarer species. And so that's what we did. And so we wanted to use this idea of embryo culture, so harvesting the immature seeds, and figure out if we could perfect the timing on that. And so what we did was a study where we collected seeds various times after pollination and basically just sowed them out and monitored them to see what happened. And you can see on this slide, it shows the dates where we collected seeds of paramina and lacera, how many plates we sowed, and this is what an experiment like that looks like. We basically put the seeds on these petri dishes like you see in the image. We store them at room temperature, and then we look at them every week or 2 weeks and basically determine if they're germinating. And then we quantify germination based on different stages. So orchids have this very different seed germination development that they go through that's quite different from other types of flowering plants. And so we were able to observe these Platanthera seeds and then determine not only if they were germinating, if we had the time right, but also if they were developing too. And you can see the scale that we use there. And so basically the way this works is, just kind of giving you an overview. We work to get permission to work with these plants, harvest the immature capsules at various stages, and determine the right stage, see if we can get germination. And then we have those seedlings, that we can use to learn how to grow or for restoration, et cetera. And what we found was with paramina that right around, let's say 25-ish days after pollination, we were able to get really great germination. And we could get germination later into up to about 30 days. But if you've ever seen Platanthera seeds, they're charcoal colored, they're very dark. And once the seeds, which start white and then transition to this dark color, once they start to get any of that dark coloration, they will not germinate for us anymore. And so we identified this appropriate timing where we got germination, and germination was really quite variable depending on which plant we harvested the seeds from. So from 5 to 91% on a particular tissue culture medium that we're using. And we got a mean germination of 53% across 6 plants that we were harvesting seeds from. And you can actually see the seedlings growing in those test tubes in the image right there. So this is a species for which there are no reports in the literature of successful germination, let alone growing them to the point like you see there, where they're big enough to be able to withstand growing outside of the test tube. And so just this past October, with Kelly Sitch from DCNR and Andy Brott from the Game Lands, we went in Dauphin County, we set up a experimental restoration planting of Platanthera paramina using plants we propagated from this known site, just again to see if this can be a successful way to restore orchid populations, and maybe even serve as a model for people wishing to do this on private properties and things like that. And so we did 2 plots with 3 transects each, and planted about 30 seedlings out. So fingers crossed, we'll see how they do next year, but hopefully in a year or two, some of these will be flowering and contributing to the seed bank and the seed dispersal of this species. So, where do we go from here? I've talked a lot about how we grow things from seed. And so I mentioned, taking these protocols and adapting them to other orchids. That's really important. And with things like Platanthera is really, again, the transition from the laboratory to the greenhouse or the nursery is still an area where we need to learn better practices for increasing the survival of these plants. And so one thing we've been doing is seed packet baiting. So basically you see those packets in the image there, those have orchid seeds in them. And then in certain places where we have permission, we bury those packets next to the parent plants where we collected the seed, with the idea that the mycorrhizal fungi that associate with those adult plants and living in that the soil or the substrate where they're growing will infect those seeds and cause them to germinate and grow in a more natural way compared to what we're doing in the lab here at Longwood. But what we're also doing at Longwood is taking the fungus that we harvest from some of these orchids, and we're growing it in the lab too. And so what I've been talking about is germinating and growing orchid seedlings without fungus, but we can also use this fungus to germinate the seeds in culture and grow seedlings that have this fungus that they probably need to survive long term already linked up with them. And this allows us to do many different kinds of studies. And so we just, a couple years ago, in a paper we're about to publish is a research project we did in conjunction with the Smithsonian Ecological Research Center. And basically, we looked at 2 species of native orchids, Goodyear tessellata and Platanthera blephariglottis, and we grew them asymbiotically without fungus in our lab, and then we grew them to the stage where they could be planted out, and we planted them into 4 different substrates, and half of the plants got fungus and half didn't. So this was all preceded by collecting roots at Beaumont Bog up in Luzerne County, where we are permitted to work. We were able to isolate these fungi and then send them down to Cirque, where they grew them on these sorborods that you see in the image. And these are basically cellulose plugs. And so we had this idea that this could be a way where we could introduce this known orchid fungus to these orchid seedlings. And so we planted the orchids in 4 different potting mixes. All of them got a sorborod, half with inoculated with fungus, half without. And then we collected data on them. We collected morphological data such as, how many leaves did they form, leaf area, number of roots, root mass, fresh weight, things like that. But then we also collected samples of the substrates and used real-time PCR, this is what they did at Cirque, to determine if that fungus was also growing in that substrate too, and whether the quantity of fungus in that soil contributed to the survival of these orchids. And that's exactly what we found, was that, the more fungus that was in this substrate, the better the orchids did. And you can see actually one of the Platanthera blephariglottis we grew from the bog, you can see flowering there, turned out to be Platanthera bicolor, the well-known hybrid from that. So we're using this idea quite a bit, and really we see this as a really great area for more research into native orchids. And so this will become more important because really what we started in Pennsylvania with a small project working on orchids, but now we're working across the state, across the mid-Atlantic, and really across the country. We're seeking to develop a seed germination and seedling development database for all roughly 220 North American native orchids that occur north of Mexico. And we're well on our way to doing this by working with our great partners here in Pennsylvania, but also across the country in places like Florida and Texas and Georgia and Wisconsin, and even as far as California. And if I can step aside from native plants just a minute, we're even taking this information that we're learning about how to grow these things and working with partners in Vietnam. And Longwood Gardens still does international field work, and we've have a long-term project in Vietnam where we're cataloging orchids there and helping Vietnamese partners learn how to grow them in cultivation, because Vietnam has, and it changes every day because there's new taxa constantly being described, but about 1,400 different kinds of orchids. And they're really concerned about, the status of some of these species in the wild. So we've been doing field work to go look at them. And then they've been learning how to cultivate plants in ex situ collections and develop what we're calling near situ collections, so that we're hoping to help them take, and they want to take some of the pressure off of the wild collecting of orchids in Vietnam, which is a really huge problem. I think much more so than here, because what is happening in Vietnam is that many orchids that occur there are used in traditional medicine. And so in Vietnam, but also Chinese people are coming across the border into Vietnam and harvesting orchids. And if you go to one of the markets, such as the one pictured here in Cao Bang, you can find orchids for sale, not only for their beauty, but also, as you can see, stuffed into rice bags and sold by the kilo to go to, be used as medicine. And so there's big concern about certain orchid species being extirpated or just becoming outright extinct because of these practices. So, by, taking some of this information we've been able to develop and funneling it to them, we're hoping they can combat some of these issues that they're facing there. And sort of as I wind down here, that talks about, that was an overview of what we're doing at Longwood. I wanted to talk about gardening a little bit because I'm an avid home gardener. I think many of the people listening here might be home gardeners, and I think everybody needs to be growing more native plants in their gardens. It needs to happen at Longwood, and it's something I take very seriously in my own garden, and I probably take it a little too far because I love the native plants of Pennsylvania, but I also love the native plants of the eastern United States. And so one really rewarding way I have found to garden is by building a bog garden and really creating a permanently saturated acidic environment for growing things like Sarracenias, and that includes Sarracenia purpurea, the purple pitcher plant, which is native here in PA, but also species from the Southeast, sundews, and it's a really great place for growing native orchids. And so I've listed some other species here, and I've also created a blog about how to do this. So if you're interested in this kind of thing, you can read the blog on Longwood Gardens website and get a better idea of how to do it. But it, again, is a really rewarding way to garden. It's a lot of work up front, but the results are long lasting, and you are able to grow these plants that you couldn't typically grow just in your average garden soil. So things like pitcher plants and threadleaf sundews, but also things like Platanthera ciliaris, the orange or yellow fringed orchid, which is one of the Platantheras that's actually fairly easy to propagate and is becoming more available on the market now as ethically propagated seedlings, but it's hard to beat that color or that plant in late July and August when it flowers. And also things like calipogons, grass pinks, which is not super rare in Pennsylvania, but this does so well for me in my bog gardens that it reseeds quite abundantly. And I have both the white flowered and the pink flowered form that you see here. And the white flowered form actually seeds more than the typical pink one, which is really kind of unusual and interesting. But this and things like rose begonia, Pogonia ophioglossoides do really well in these types of gardens. And I think these types of gardens really, I know with my own children and other people I have to my garden, they're just great educational tools, and that people are naturally drawn to the kinds of plants that grow in there, and that it can be really great, I think, for educating people about, not only the importance of native plants, but the diversity of native plants, and that we have orchids in Pennsylvania, because I think many people are unaware that orchids even grow in Pennsylvania. So That pretty much wraps up my presentation. Like I said, we're doing a lot of work here, and I don't do all of this alone. And we have a really great list of partners and people and places that we work with. And I mentioned some of them here. I specifically want to call out my team here at Longwood that does Ashley Clayton, who does a lot of the work at Valmont Bog and is really doing great research on that project. I have a team of volunteers. You can see Ed Leinbach here that work and basically do all of the seed sowing that we're doing. And these guys are retired PhDs from the pharmaceutical industry. So we've been able to learn a lot from them about lab technique and how to refine some of the things that we're doing with the orchid. So many people have helped and aided in this. And Kelly Sitch in particular has been a great contact and person to work with regard to many of these orchid restoration projects that we're doing in Pennsylvania. And so thank you to the many people that have sent seed or come to Longwood and been on a tour or helped in a project in some way. So thank you very much. And thank you for having me speak at the symposium.
Thank you so much, Peter. That was fantastic. So we have quite a few questions here. I think we'll run a little bit into our break. So we'll take questions for the next 5 or so minutes. And then we'll give just a little bit of a transition to the poster session. And I'm sure people can, still take a little bit of a break if they need to during that poster session. So Peter, one of the questions, you mentioned that nearly half of the orchids in Pennsylvania are rare or vulnerable. So what is the greatest threat to local orchid populations? And then also, what are the success rates for the kind of restoration work that you're doing?
Sure, good questions. And so I think that, it's a combination of factors that contribute to orchid rarity. I certainly, in my region of the state, we have 40 to 50 white-tailed deer per square mile. I think the ecological threshold is like 4 or 5. And so, things like lady slipper orchids, like these really small local populations we have, they flower and set seed, but the deer eat the seed capsules. And so, I think that the deer are having a really bad impact on native orchids. But I also think habitat loss, I think broad scale application of herbicides and, like on roadsides and in power line right of ways, especially in the southeastern United States, is a huge problem for orchid diversity, because those are, areas that are maintained as like constantly disturbed. So you get pitcher plants and sundews and orchids and things like that in some cases. And so I think that's really detrimental. And then, just habitat loss, so I think that's sort of the trifecta of that. And with regard to our success of putting orchids out, we haven't done many studies. We've done one with Goodyear tessellata. That one didn't do very well, but what happened was there was fall of big trees in the habitat where it was growing, the white pines, the Pinus pungens that it was growing under. And so what had been a very dark sort of, cathedral canopy with this, understory of lycophytes and orchids and things has changed quite substantially. And so after a few years, I think 4 of the plants are still there out of about 80 that we planted, but I think that wasn't the best trial. We've done these others and we're sort of waiting to, see what happens. And, I think it's going to vary a lot between species. I think, as mentioning, cypripediums appear to be very long-lived, but something like spiranthes appear to be very short-lived, so I think it really depends on the species, and there's not a lot of research about that. So it's something we really want to do a lot more of.
That's awesome that you're still learning a lot about the demography of these species as you're doing this kind of like what we usually think of as a next step.
Sure. Yeah.
So another question, are you doing anything to try to augment the genetic diversity in your restoration population, since we know that many of these are very small and might have low genetic diversity?
Well, that's our hope. And that's really, I think, something we want to move into is we don't have, molecular capabilities here at Longwood. And so, we're seeking partners that can sort of do that work, looking at, these small populations and, what we're cultivating and learning more about that. And it's something I hope we can move into ourselves within the next few years. But that's, that's something we really need to figure out. So yeah, more to come on that.
Do you know if any of the declines that are being seen in the Eastern North American orchids are because of a loss of pollinators?
I think, I mean, anecdotally, I think that is happening. I know like one, we work with DCNR partners at the to monitor a population of Arethusa bulbosa, and we started hand pollinating those a few years ago because they weren't setting any seed, and so the plants are in sort of a closed-in habitat. Maybe the pollinators weren't finding them, maybe they weren't there, but it's been pretty much the case where the only seeds that are sitting there are the ones where we're hand pollinating. So, and I think this has been, documented better by others. It's something I wish we could do more of, but I think that, pollinator decline is an issue we need to focus on more, especially for orchids.
And with the different Cypripedium that you're planting back out, have you seen any of those reseeding or
Haven't
even
seen
having
them
a viable seed?
Yeah, not yet. Well, the first ones it takes about 4 or 5 years to get them to flower from seed. And so we are seeing some seed set on the first ones we've grown, but like for these restoration studies, these are still young plants. And so, we plant them before their flowering size, but, hopefully there'll be some seed recruitment, some pollination and seedling recruitment. So these are very long-term studies, and so it's hard to assess exactly what's happening, I think, in just a couple of years.
So then we have some folks who seem pretty excited about your idea of bog gardens. Do you have recommendations on where people can purchase those kinds of plants?
There's a lot of online sellers, that produce plants in ethical ways. Like there's a place in Maryland called Carnivorous Plant Nursery. There's a really great place called Meadowview Biological Research Station, which is in Fredericksburg, Virginia. And all of the proceeds that that come from selling plants goes to the restoration of a site called Joseph Pines, which is basically the last natural population of Sarracenia flava left in Virginia. So they're taking all this money and doing this really great conservation work. So they are highly recommended. And there's a lot of them. Those are the two I'd recommend off the top of my head.
Awesome. Thank you. So Peter, there were quite a few other specific questions for you about, sort of the details of your talk, and also some folks who are excited to maybe collaborate or suggest other collaborations for you. So I'm going to let Moira pass those on and with respect to time. And so just thank you again so much. This was really fantastic to hear about your work with orchids finally.
Great. Thanks for having me.
I appreciate your time. So what I'm going to do now is I'm just going to transition us to some slides that sort of give directions for where we're going next in terms of the poster session. So we've had 28 students who have submitted posters, so that's just really fantastic. So all of our students were either undergraduate or graduate college students, but we do accept high school submissions, just in case a precocious high schooler who might want to submit for our next symposium. We would love that. So again, with the poster session, this runs until 3. If you need a break, please do take a little break sometime during the poster session, since we didn't give you much time now. And when you're in the poster session, when you're in the poster platform, there is a place in comments where you can go ahead and give comments to the students, and then they will respond back to you. You do need to press the refresh button for the posters because this specific platform, it's not sort of automatic like it is in Zoom. So here underlined and in black is the link that you need to go to view the student posters. I recommend that you open it in the Firefox internet browser, or you can also use Control plus the scroll on your mouse to kind of see a bigger size of the posters. It's a little bit cumbersome if you're using Chrome. So for me, I actually downloaded PDFs of posters I really wanted to see so that I could zoom in and out with a PDF reader. So those are some tips. Again, please do talk with our students. Remember to refresh your page. If you are a judge for the posters, Emily will be waiting to hear from you via email so she can tally up those scores. And we do have Sam, who was from Juniata College. Her poster was submitted late, so we didn't get it uploaded to the regular poster platform. At least it wasn't when we got started here at 1 o'clock. So you may need to go to the link that's posted in the comments by her name to find that poster. So again, please join us for the poster session at this link, and then come back to this Zoom room at 3 o'clock to learn about all of the cool botanical finds around the state. Thanks, everybody. Just put in chat or feel free to email Moira if you're having any problems with this.
Hi, everyone. I hope you had a great opportunity to view some of the great research from the student posters. And apologies to all those who had some confusion on the comments. From our hosts here, we do have sincere apologies, and we'll talk to the platform providers about that. And so I wanted to also thank you to the students from the poster session for stepping up and putting yourself out there for this session. And I look forward to seeing all of you in the future as your research continues. And I wanted to thank also the advisors who pushed and encouraged your students to participate in this poster session. I know as a student, sometimes it's a tough thing to do. And lastly, I want to offer a huge thanks to Emily Rawlinson, a PA Botany Symposium committee member and faculty at East Stroudsburg, for facilitating and organizing and pushing this poster session through. And to the judges also for interviewing and asking critical questions on the work that was presented. So stay tuned. After the end of Cool Finds, we will announce the winners for each category. Okay, and now on to Cool Finds. Okay, Cool Finds, for those of you who haven't been to a PA Botany Symposium before, are submissions from botanists in the field on unusual, exciting, or otherwise noteworthy botanical discoveries recently in Pennsylvania. And because we haven't met as a group for probably 3 years, we have accepted submissions from 2019, 2020, and 2021 from the field. Before we get started, for those who don't know, I'm Chris Firestone. I'm a founding committee member of the PA Botany Symposium and a botanist with Pennsylvania DCNR Bureau of Forestry, which is the state agency in Pennsylvania with jurisdiction of Pennsylvania's native wild plants. So on to Cool Finds. So what is going to be presented today is what has been submitted. You know, there's a lot more out there. This is what was submitted when we put out a request for Cool Finds. I didn't confirm the IDs on these, so if you see something that is not identified correctly or you have questions on it, take it up with the collector. I did, however, check on the state statuses, so those should be current. I hope that most who submitted these records for Cool Finds are joining us today and can contribute in the chat as questions come up. There are 32 records to cover in a short period of time, so there will be not a lot of time for questions. Nomenclature follows flora of the southeastern United States and Pennsylvania. Pennsylvania edition from October 2020, which is by Alan S. Weekly. And sometimes it is different than what we list on our Pennsylvania Natural Heritage website. So I have included that too. I'm attempting in the next 40 minutes to tell a story about each cool find, but because we are constrained by time, it is going to be a short version, and it's what I know. But just like every story, there's another version. So I encourage you all to reach out to the botanists and collectors who submitted that cool find to find out more of the story. And also, lastly, I hope that you do get to check out the link for poster 22 from the poster session from Sam Vitar at Juniata College on virtual cool finds. We did do a little bit of collaborating here on this, but there are a lot of, from her poster, there are a lot of cool finds that were not submitted, that maybe not everyone knows about this awesome way to present your cool finds. But please take a look at that. It has some very interesting information on that. So the categories that we will discuss are newfound occurrences of Pennsylvania endangered and threatened species, newfound occurrences of S1 to S3 communities, species that are new to Pennsylvania, and otherwise amazing, enlightening, or alarming finds. And not to be to spoil any stories, but at this time, there were no alarming finds submitted. So whether that's good or bad, I'm not absolutely sure, but that'll be determined. So I do want to give a thanks to all the indigenous people in Pennsylvania who took care of the land and plant populations before there were botanists who were out there documenting the wonderful plant species in Pennsylvania. And thank you to all the botanists today who work in plant conservation to document, study, and conserve over 2,500 species of native wild plants in Pennsylvania. This map just represents a snapshot in time of some of the native people in Pennsylvania, and my apologies to any tribes or peoples that are not represented on here. So the 32 cool finds presented today are very diverse, just as they are Pennsylvania is diverse. They represent 20 plant families, 20 counties, and 14 botanists who were brave enough to submit their finds for today's presentation. If you count the locations for today's finds here on the map, you'll see that there are not 32 because there are many species from that same county. So there's more than one sometimes from one county. Okay, so let's get started. Our first category is the newfound occurrences of Pennsylvania endangered and Pennsylvania threatened species. These are ones that have been documented before in Pennsylvania and have a status of Pennsylvania endangered or Pennsylvania threatened, but these are just new occurrences. Maybe some of them are new counties. Just for those of you who are not familiar, each location, each species will have a geographic range map and then an up close one of Pennsylvania. And these are from BONAP. And just wanted to let everyone know that yellow means that the species is present in Pennsylvania, but rare. The light green indicates that it is here and not rare. And sometimes you will see an orange for the county, meaning that it is no longer found in that state or that county. Okay, so our first one is Albicuris aqualis. And this one was submitted by Scott Lynn Martin from this year. He found it in Snyder County. As you can see from the map, that is a new county record. And it, in this case, he did find it in a cornfield wetland. So it was in an agricultural use area. The next one is Bartonia paniculata, subspecies paniculata. Here in Pennsylvania, that is a state, has a state status of Pennsylvania rare, a global rank G5, T5, and a state rank of S3. And again, just a little explanation. Wanted to let those who are not aware of the NatureServe methodology that the G rank and the S rank are based on methodology from NatureServe. And this is what our Natural Heritage Program uses also. The state rank here of Pennsylvania Rare is set by Pennsylvania DCNR Bureau of Forestry in our plant program. The Bartonia is spruce stem Bartonia. It was found by Scott Martin in August of this year in Schuylkill County, the Swatara Creek watershed, in organic muck and sphagnum moss. This is a new site, but it is not a new record for Schuylkill County. The next one, another a grass, is Casmanthium latifolium. This is a tentatively undetermined, currently a tentatively undetermined species, and it is globally ranked as 5, so it's secure, but it does have an S1 state rank. It is called wild oats, river oats, fish on a stringer. I just added that because Weekly had that as a common name, and I just thought it was a very cool common name. So I wanted to add that in there. And again, this was found by Scott Martin in September of this year in Lancaster County. According to what he submitted, it is along a railroad on bedrock outcrop. I'm not sure, you can ask Scott a little bit more about it, but he did say it's very, it's also very close to the Maryland border. Hopefully that'll keep moving along. I live in an area where there's very slow internet. Okay, our next one is Hypericum stragulum. Again, it was submitted by Scott Martin in the same area as the previous one in Lancaster County. And this one, of course, you can see is the actual bedrock outcrop along the railroad. And again, it's not a new county record, but it's a new site. And I have, for the BONAP maps below, for the distribution maps, you can see I did add Hypericum hypericoides submuticale because that is the way it is listed in BONAP at this point. So you have to do a little bit of finagling when you're in BONAP to find your actual species sometimes. And again, Scott was very busy this year. As always, he's a great contributor to species. And this one's Chester County. Again, it's a new occurrence in the county, not new for the county. Woodwardia areolata. And in Weekly, it does have a different genus, Lawrenceria areolata. This is netted chain fern, and it is currently proposed in Pennsylvania as a PR, but it does have a global rank of G5 and a state rank of S3. Again, if any of you have questions, want to know a little bit more about this, please reach out to Scott. I don't know if he's on here, but hopefully he is. Okay, and here's an interesting one. I know this was one that Peter had maybe not mentioned, but he did have a picture of some of the seeds germinating in a Petri plate. And this is Three Birds Orchid. It has been kind of on everybody's plate a little bit. It's kind of a small one to see. It's not big. It's not a big and showy orchid like the lady slippers or some of the plantatheras that Peter was talking about earlier. But we did have 2 submissions for this one. Bonnie Isaac and Joe Isaac from August this year had submitted this one in Forest County in the Allegheny National Forest. And according to Bonnie, this has expanded the record from the year before. They had found a population, but this one that the record was a little bigger. It expanded from the year before. And Stan Galenti, I had seen him comment earlier today, so I hope he made it back from the poster session, submitted one for Luzerne County, also from September of this year. And that is a new county record. And according to Stan, he said there's about 300 plants at this site in this population, which is sounds like a great one to check out. And this one was submitted by Norris Muth, Listera smallii. Also in Weekly, it is identified as Miatia smallii, kidney-leaved tway blade. And I'm not clear on this, if this was one that they had found through their virtual, looking through their virtual scan of records in iNaturalist. But this is one among bryophytes in a riparian corridor that they found in Huntington County. Again, I believe it's a new site. However, it is not new to the county. Panicum longifolium. It is a Pennsylvania, tentatively undetermined species. It is global rank of 4, but a state rank of 1, long-leaved panicum. And again, this was submitted by Stan Galenti from July of this year in Luzerne County. And again, as you can see, this is a new county record. And he did find this in Petey Sandy Vernal Pool Edge. This is a pretty cool one, not to be confused with James Rocket by any means. Iodanthus pinnatifidus, and this is also known as purple rocket. It's Pennsylvania endangered G5 S1 species, and it's found by Joe and Bonnie Isaac in May of 2019 in Washington County. It's not a new county record, but it is a new site. And the information that Bonnie gave me when she submitted it, it was east of Clarkville. And I did want to mention, I didn't get to mention it before when I mentioned the poster by Sambatara Juniata College, that many of these submissions are in iNaturalist too. I know Bonnie and Joe use iNaturalist quite a bit for their species finds, and it was easy to get pictures because they were all right there. So if you haven't started using iNaturalist or just want to take a look, I'd encourage you to go ahead and start doing that. Okay, and here's another one by John and Joe and Bonnie Isaac, Madalea obliqua, and it's called angel pod or northern spiny pod. It's in the dogbane family. They found this in June of 2019 in Greene County. And this is a new county record. That's always exciting when we have new county records. And again, this one is north of Clarkville. Oh, this is a beautiful sedge, and I'm sure it's very easy to identify. Golden fruited sedge, Carex aurea. And again, this is Bonnie and Joe Isaac from July in 2019. This was Erie County, south of Harbor Creek, and it is a new site, not a new county record, but it is a new site. And as you can see, when I down at the right-hand corner, I say see next slide, because sometimes botanists get very lucky when they're out at a new site and walking around. And so on this day, also at the same area, they found another Pennsylvania endangered species, Equisetum variegatum. This is a Pennsylvania endangered species, G5S1, and it's called variegated horsetail, and it's in the horsetail family. And I think I covered this one. This is a fun species to find another location for. This is Heuchera alba. It does, you can see it does not have a Pennsylvania status yet. However, NatureServe has said it's a G2 species, and we have given it an S1 state rank here in Pennsylvania. There's a lot of work being done with this. If you haven't seen this yet, Chris Martin's lab at Biennial has put together a cool finds video, and it's on YouTube, and they're talking about how they actually came across this species. I believe that was in 2017. And in 2018 at our Botany Symposium, we had Chris present all the social media that went into the finding the species and how it was eventually identified when it was misidentified, but somebody on Instagram had recognized it. And so we do have a new species in Pennsylvania for this, but it's presented today in this section as a new location that was found by Joe and Bonnie Isaac in May of 2020 in Huntington County. The original population was found in Union County on the Shikellamy Bluffs. And that's, you can find all about that in the YouTube video that was put together for that. And again, this is a new record for the county. I believe there's more field work going on, looking, more bluffs for this, rocky areas. And eventually there will be a proposal put in to give this a state status once more of the work, more of the field work has been done. Okay, this is a very, very cool, fun species. And this is Viola glaberrima. It was proposed for Pennsylvania endangered in 2020. It does have a G5 global status, T3, and state status of S1. It's the northern wedge-leaved violet. And it was found in different places, again by Bonnie and Joe Isaac, in May and August of 2020 in Westmoreland and Indiana counties. I didn't get the Bonap map for it because it's not even listed in Bonap. And Westmoreland and Indiana counties at this point are the only counties that perhaps we know about at this point. But the very cool thing about this one is that there was a record from 1920 that this species was here, and it was never documented until Bonnie and Joe found it. And Moira, if let's listen to Bonnie's story about this. I don't think she knows that I put this into the Google Find, but here's Bonnie's story about finding this.
Hi, I'm Bonnie Isaac from Carnegie Museum of Natural History. I'm out in the field looking for some Viola glaberrima, a species that hasn't been seen in Pennsylvania since 1920. Believe I might have just found it. See if I can show it to you here. See that yellow violet? Think we might have just found a good one. There are at least 3 other species of violets here on this slope up above the dam. I'll be sending some pictures of this to the violet expert Harvey Ballard and see if he confirms my suspicion that this is the thing we've been looking for. And if it is, it's been 100 years, but 1920 to 2020, it's all good. Okay, so I heard back from Harvey Ballard. He says we nailed it. We did find Viola glaberrima. And so that's one more extirpated plant from Pennsylvania that is now extant. It'll automatically go to endangered.
Thank you for sharing that. Oops, now I need to move on again here, sorry. All right, thank you, Bonnie, for sharing and telling the story for this one. It's such a cool story. And as I see somebody commented that we love that you're on TikTok. If we could all be on TikTok and tell our botany stories. Okay, and the next one is Isotria medialoides. It's small world Pogonia. And as you can see for the status on this one, we have added the federal. This is a federally threatened orchid species. It's Pennsylvania endangered, G2, G3, and the state status is S1. We do not find this species often here in Pennsylvania. Some of those species, counties that are highlighted in yellow there are probably historic records. But Bonnie and Joe in June of 2020 found the expansion of the existing population in Center County, which was kind of exciting, by a half mile. And because of the rarity of the species, of course, when it goes into iNaturalist, I believe all of the records do, you do have the option of offsetting the location point. I'm not an expert in iNaturalist, so don't ask me those, but I'm sure Bonnie can help you with questions using iNaturalist. The next record is Trichostoma cetaceum. It's a Pennsylvania endangered, G5S1, and it's called narrow leaf blue curls. And it is in the mint family. And again, that's another record from Bonnie and Joe of September of 2020 in Fulton County in Shale Barrens. And this is a new site and a new county for this record. And again, as you can see, we do have the next slide joining these because at the same site is another exciting species find, Galactia regularis, and it was listed, well, it still is listed until the regulations go through as PX, which means Pennsylvania extirpated, G5 and SX to go along with the extirpated state status. So that at one point we had historic records, and you can see on the Pennsylvania map there that the counties that it was historically found in are orange. But Bonnie and Joe found this in Fulton County at some shale barrens. And again, this was a record from 1941, and they have rediscovered this one almost 69 years later. in 2021 at a vascular plant technical committee, it was proposed for PE and will go forward in our next regulation package for Pennsylvania endangered. Our next one is Hylotelephium telephoides. It is a PR G4S3 species. And in weekly, it is known as sedum telephoides. And the common name is Allegheny live forever. This is one of our native sedums that we have in Pennsylvania. Again, it's from Bonnie and Joe in September of 2020 in Bedford County, north of Town Creek. And again, it was last recorded in 1941, but it is another shale barren species. And they were lucky again at this site and found another record hanging out there in the shale barrens, the same place in Bedford County, north of Town Creek. Trifolium virginicum, and this is a Pennsylvania endangered clover, Kate's Mountain clover, or shale barren clover, Pennsylvania endangered G5, S1. And as I was doing a little bit of work and finding out a little bit more about this story, I did find out that this was found, I believe, in 1892, the species on Kate's Mountain in West Virginia, hence its name, Kate's Mountain clover. So sometimes the common names are there for a purpose. Well, they're always there for a purpose, but the common name for this one is describing where it was found. This species has been in cool finds before. This is another location for this one. This is Goodyear tessellata. And it kind of has been being searched for back in maybe 2019. And if Mark's on here, I apologize for not having my dates right. But 2018, 2019, Mark Simonis has been finding it in Tioga County in many of our 100-year-old or pine plantations planted on the state forest. And in this year, in July, Joe and Bonnie again found it in Forest County. And this is a new county record on the ANF. And this one, oh, and I believe she said that they found this one last year too. So, so it is now, because of the new populations in Tioga County and the ones in Forest, it is now proposed to be downlisted to Pennsylvania threatened instead of Pennsylvania rare, based on the new populations that we are finding. This species does come up a lot in the rare plant forums and the vascular plant technical committees, Erigenia bulbosa, and it is currently listed as Pennsylvania threatened and here in Pennsylvania and G5 and S4, but it is proposed, it was for PR, both for the east and west populations. So the common name is Harbinger of Spring. And this is a new county record. As you can see from the distribution map there, the range map, there are most of the counties in the west do have this species, but this one was found in Venango County on Clear Creek State Forest along the Allegheny River. So this was definitely a new record. The State Forest has been notified, and hopefully we can put some protections on the State Forest for this one. And here's one that has been, that was mentioned again by Peter, another orchid. I don't know. I hope you guys are counting up these orchids that have been in this cool finds presentation. But this one's Cypripedium parviflorum, PE G5T3T5, and it's an S1 or S2. And if you can't see, that is the Cypripedium parviflorum variety, parviflorum. And this one was from Center County, from Bonnie and Joe. Our next category is the newfound occurrences of S1, S3 communities. And there's only one that was submitted, and Jackie Braun she works with the Pennsylvania Natural Heritage Program, and it is pitch pine rhodora scrub oak plant community. It's in Long Pond. The purple plant that you see is the rhodora, the Rhododendron canadense. And this one is this small plant community has been protected by the Wildlands Conservancy. Our next cool find are new species in Pennsylvania. This was another exciting one. And this is, as you can see, it's a new species in Pennsylvania, Amelanchier amabilis, the lovely shad bush. We don't have a state rank here in Pennsylvania. New York doesn't have a state rank either, but it is a G4 question mark. And this one was found this year in May by Mark Simonis and Bonnie Isaac and Joe Isaac in Tioga County at Colton Point State Park. And the interesting thing about this one, it is found in some of the overlooks from the Pennsylvania Grand Canyon. And as you can imagine, those overlooks are maintained to keep vegetation out so that the public, when they're coming up to the Grand Canyon, can look out down through the canyon and the valley. And it just happened that Mark and I were there this year checking it out this summer, and there was a crew that was cutting the vegetation this year. So we, just in the nick of time, managed to save most of the plants that were right there at the overlook. Here's an interesting one. I hope you got to see the poster session for this one. This is Allium burdicii. It's a G4. Again, it hasn't been given a state status yet. I know Eric and Sarah and Cassie are still really much working on locations. I believe I just found out that there were at least 4 locations for this species here in southwestern PA. And this is kind of interesting. This is kind of exciting. It has a long story to it. I don't want to make it any longer, but Eric wants to thank all the regional botanists who suggested sites. And if anybody has any other sites where they might find the burdicii, please let Eric know about this. The leads are going to be much narrower. And here's a new one. This is kind of a very interesting one, Desmodium sesquifolium. It is PX, but it was submitted by Stan Galenti from Luzerne County this September in 2021. I know for sure I would like to hear more about this. It's in the P family, sessile leaf tick trefoil, or trefoil, sorry, tick trefoil. So if anyone would like to know more about this one, please reach out to Stan for his story. And one more, I have a couple more cool stories. How do you make an entire species extirpated in Pennsylvania? Well, you just dig it out. That's Steve over there digging out Marshallia pulchra. The interesting story on this is this used to be known as Marshallia grandiflora. Barbara's buttons, it's still known as Barbara buttons, but a recent paper by Wesley Knapp, Poindexter, and Alan Weekly have determined that Marshallia grandiflora is now extinct in Pennsylvania, extinct in its range, and it is the 53rd extinct species in North America. So in Pennsylvania, we have delisted Marshallia grandiflora, and our Marshallia grandiflora records are now Marshallia pulchra, which are PE G3S1 as proposed. There is a paper that they put out on these, and I believe that There is a link. I don't know if I gave more of that link or not, but there is a link for this one if you're interested in reading more about this. So I'm just kidding when I'm saying how do you make an entire species extinct. The interesting thing is that the holotype for this species, from Marshallia pulchra, is now from Fayette County in the Youghiogheny River. So the holotype for the species is in Pennsylvania. So that's a very cool story on this one. And our very last category, I know I'm running out of time. I didn't think I was going to do this. Another orchid, Spiranthes lucida. And this one was submitted by Juan Serter Herrera from June of this year in Center County along in Black Moshannon State Park along the boat ramp. But also Joe and Bonnie have submitted this from Mercer County, which is a new county record. A beautiful purple milkweed with all the news about pollinators and the monarchs and the milkweed. We do have a purple milkweed here in Pennsylvania, Asclepias purpurascens. We don't have a status for this, but it is a G5S4 status. Thank you, Craig Hall, for submitting this one. We don't see it very often. This is from July of 2020 in Adams County. Hastiola suviolens, and it's also known as Senecio suaviolens. Does not have a state status, but is a G4S4, sweet scented Indian plantain. Jason Reed submitted this of October of this year from Venango County. It's nearly a monoculture, he says, near a large creek. And I believe this is our last one, Theoglossum vulgatum. Again, there's a big story on this one. It is extirpated. Scott Martin submitted this one from May of 2019 in Indiana County, a rich deciduous forest, southern adder's tongue. So I'm sorry, there's one more. And Ranchium aquaticum, it's a PR, and it's a G5 S4. It's in the Ariaceae family. It's the same as Jack in the Pulpits and skunk cabbages. However, this is a new county record, I believe. Okay. So that is cool finds for 3 years in a row. Okay. Now on to, I know we don't have any questions or any time for questions at this point. Okay, student posters. Is everybody ready? Autumn, do I have control of these?
I have control of the slides. I'll proceed when you tell me to.
Okay.
Just one moment, technical problem. Autumn, we see your presentation notes screen. We're not seeing the presentation slide.
Okay. Okay, so we will start with our student poster awards. We do have 2 categories, undergraduate and graduate. And those places, they do get a monetary award for first place, $200, and second place is $100. So without any more hesitation, drum roll, our undergraduate winners, for second place is Sarah White from the University of Scranton, quantifying geographic variation in flower color in Mimulus ringens using citizen science data. Congratulations, Sarah. And our first place for the undergraduate is Diamanda Zizes, and I'm sorry if I pronounced this wrong, from Bucknell University. What happens when you cross-plant species with two distinct sexual systems? An ex situ hybridization approach. Congratulations, everybody. And our graduate winners, second place is Cassie Stark from Penn State University, a habitat comparison between two ramp species, Allium trichocum and Allium burdickii in Pennsylvania. And congratulations, Cassie. I'm sorry I didn't have much more time to talk about the two differences, but it was a great poster too. Thank you. And first place graduate winner is Cheyenne Moore, University of Pittsburgh. Invasive hybrid vigor, niche and trait variants between 3 knotweed taxa. Great job, everybody, and congratulations to all 4 of you. All right, I think it's back to Autumn. Thank you, everybody.
So thank you all so much for being here with us today. I haven't participated in too many virtual symposiums, but I'm always really nicely surprised at how engaging I end up finding them. I really enjoyed today, and I hope that you did as well. Before we go, I'd like to thank Peter Zale, Chris Firestone, and all of the students who submitted posters for their time and for their wonderful content. Thank you all for participating in the PA Botany Symposium with us today. I know it can be tough to give up a nice Saturday afternoon to be on the computer. On behalf of the Pennsylvania Botany Planning Committee, we hope that you will view more of our content online at pabotany.com and join us next year in person for the symposium at the Penn Stater. At least we don't all have long drives to get home today. Goodbye, everyone. Thanks.